CRISPR is this fascinating, powerful technology. It's got a very clunky name. It's called clustered regularly interspaced short palindromic repeats. Now, what does that mean? So the name actually refers to the way it interacts with DNA. And it's a way to manipulate DNA, to edit DNA in a way that previously is much more powerful than previous methods, much simpler, much cheaper, and the important part is it's exceptionally precise. So as you probably know, our book of life is made of DNA. DNA itself is many millions of base pairs, which is like a language. And within that language, there are certain regions which code for genes, and those genes are incredibly important because those genes go on to make up everything about us. There's 40,000 proteins that become outputs of those genes, and they're involved in really our health, our well-being, and any defect in those genes becomes problematic in disease. What was previously attempted with gene editing was to manipulate genetic information in blocks, basically in big pieces. It's kind of like trying to edit a book by only being able to rip out a page at a time and transfer a page at a time without really being able to control the actual words. The power of this technology, it literally comes down to the individual letters. So the precision is far better than anything that's happened before. And the excitement in the scientific community is being able to go in and very precisely make changes in DNA of actual genes that you can actually turn off bad genes, or you can potentially repair genes that have got mutations in them where the code is written incorrectly. People think that at some level every disease has got a genetic component. So monogenetic diseases are ones that we can trace back to a single gene that has a defect. Often those genes have multiple mutations, but at least it's a single gene has been identified. So the most promising application of CRISPR would be to modify those monogenetic diseases. And those monogenetic diseases, they broadly fall into two categories, kind of curiously named toxic gain of function and toxic loss of function. So toxic loss of function is the one that's kind of intuitive. That means that the gene has got a defect in it. The person loses the function of that protein, and that causes a disease. So a well-known, well-studied example would be sickle cell anemia. So a single base pair mutation actually causes a change in the structure of hemoglobin that then creates this unique sickle cell shape for red blood cells. There's another disease in which a mutation actually causes a clumping up of different proteins. It's called transthyretin, and that leads to a disease called amyloidosis, where these proteins, which normally don't stick to each other, now because of this kink in them due to the mutation, they become very sticky, they form aggregates, and those aggregates can build up in various cells in the body, sometimes the brain, sometimes the heart. That's called toxic gain of function. CRISPR potentially could be useful in either one of those. And in fact, there are companies that are looking at those diseases as well as a number of others. Another potential category is oncology. It's been well known now for some years that our own immune system has the ability to fight cancer cells and really essentially dissolve microtumors. And cancer is a clever entity, and it evades the body's internal immune system. So what that means is that the cancer becomes invisible to our immune cells. And that invisibility is due to certain proteins that are created to, as checkpoints to interfere with the immune system attacking our cells. So cancers essentially mimic our own cells by taking advantage of these checkpoints. So one of the applications of CRISPR would be to remove immune cells from the body, apply the CRISPR technology, and then turn off these checkpoints and put those immune cells back in the body with the hope that then those immune cells would clear the tumor away. One of the biggest risks of CRISPR is what's called gene drive or genetic drive. What that means is that because you're actually manipulating genes and those genes get incorporated into the genome, into the encyclopedia basically that sits within cells, potentially those genes can then be transferred on to other organisms. And once they're transferred onto other organisms, once they become part of the cycle, then those genes are in the environment. That's probably the biggest fear of CRISPR, that humans manipulating the genetic code and those manipulations get passed on generation to generation to generation. And the fear is that we think we know what we're doing, we think we are measuring exactly what changes we're doing to the genes, but there's always the possibility that either we miss something or our technology can't pick up on other changes that have been made that haven't been directed by us. And the fear then is that those changes lead to antibiotic resistance or other mutations that go out into the population and would be very difficult to control. Basically creating incurable diseases or other potential mutations that we wouldn't really have control over.